episode 287 of the Stem Cell Podcast, Blood Development, with Drs. Andrew Elefanty and Elizabeth Ng. Hello, everybody. We are Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, which is a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying this listen, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have a great pair, and someone I think all stem cell biologists recognize the names of because they've probably used a few of their tools. We're talking about Drs. Andrew Elefanty and Elizabeth Ng. They're from the Murdoch Children's Research Institute, and they're on the podcast to talk about a pretty groundbreaking study that was the culmination of decades of work across many research groups contributing and the product, a lot of the tools that they've generated and shared with the stem cell research community. Yes, they're talking about my favorite cell type, generating hematopoietic stem cells for tissue repair and regeneration. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, we'd like to remind our listeners about the ISSCR annual meeting happening June 11th to 14th in Hong Kong. Join the world's best and brightest in stem cell research and regenerative medicine for a 4-day program comprising the year's most significant new advances in the field. Learn more at isscr2025.org. We're going to talk about a paper first here, which is a very big deal here in Nature. Something very near and dear to my heart, pun intended. And we've been hearing about this, at least the folks in the cardiac field have been hearing about this for a few years now. This is engineered heart muscle and allografts for heart repair in primates and humans. And we've talked to Chuck Murray on this show about the applications of pluripotent stem cells for cardiac repair. Of course, this is a big dream in cardiovascular regenerative medicine. You know, after somebody has a heart attack, a myocardial infarction, perhaps you could use pluripotent stem cells as the source cell type to make cardiomyocytes, cardiac tissue for therapeutic applications to restore heart function after a myocardial infarction. And Chuck Murray's work has been great, and that's been ongoing for a number of years now. Unfortunately, some of the work that he's been doing has been associated with induced arrhythmia, which is, of course, a huge problem when it comes to cell transplantation into the heart. And they've done a whole ton of engineering-based approaches to modify some of the genes responsible for the arrhythmia, and they're approaching things in a unique way in terms of injecting those cells into the myocardium and seeing if they can integrate both mechanically and also electrophysiologically. And on the other side of the world, I suppose, and the other side of the field is Wolfram Zimmerman, who I know actually pretty well from my time in Joe Wu's lab. He's a close collaborator with Joe Wu and has been a really incredible pioneer in tissue engineering and cardiovascular research and tissue engineering in Göttingen. He's one of the heads of the German Heart Center, German Center for Cardiovascular Research, DZHK in Göttingen. And over the past 15 years, he's been working on these engineered heart tissues, EHTs as they like to call them, and I've seen a number of these employed in the Joe Wu lab through collaboration and various in vitro studies, but also they've been focusing on the clinical applications for these engineered heart tissues, these EHTs or EHMs, engineered heart muscles, to see if you could actually slap these guys on as almost like a Band-Aid on top of the heart to help restore some of the lost function after a myocardial infarction. At least that's the dream. This is not a long-term solution. This is not going to completely replace the heart. This isn't something like those pig xenotransplantation hearts where you're going to completely replace the organ. This is intended to be more of a bridge to help restore the function of the failing heart for a few months until a suitable replacement heart can be found, whether it's through pig xenotransplantation ultimately, or right now probably a human heart. So again, this is a bridge approach using these engineered heart muscle allografts made from induced pluripotent stem cell derived cardiomyocytes and stromal cells. So they did a really a beautiful study published in Nature just very recently. First, employing these EHMs in a macaque model. Well, actually, before that, they confirmed the in vitro and in vivo equivalents of the newly developed rhesus macaque EHMs with the prior nude rat models. So they did the rodent work first, and then created this EHMs from iPSC macaque iPSCs in a GMP approach, good manufacturing practices, because everything that they're making here is for the intended purpose of going into people. So it's a GMP compatible human engineered heart muscle formulation from IPS. Um, they transplanted these things into the epicardium, or basically slapped them on as a band-aid kind of onto the epicardium of these macaques. And the epicardium is the outermost layer of the heart, as opposed to the myocardium, which is a little bit more on the inside. And they showed really great retention up to 6 months of these engineered heart muscles, a dose dependent enhancement of the target heart wall. That's really important, an improvement in function of these EHM grafts that were constructed up to 200 million cardiomyocytes per graft. So you had an improvement in function in these macaques, and that's actually with or without the induced myocardial infarction. So after, it doesn't matter if you actually induce the MI or not, you're getting an improvement in cardiac function, and that improvement in function is measured through pretty standard means in cardiac biology, which is ejection fraction, or in other words, how well the heart can pump after receiving these epicardial band-aids, quote unquote, right? So improved contractility, which is a measure for local and global heart support. They also did the pathology, the cardiac MRI, which confirmed that the cells were being retained in the macaque hearts, in the number of these macaque hearts, and actually improved vascularization too. So that's pretty amazing. And I think a really critical point here that somewhat separates it from those original Chuck Murray studies is that they did not see any arrhythmias. And their reasoning is that they're putting these EHMs, these engineered heart muscles, on top of the heart in the epicardium as opposed to more intramuscularly like inside the heart. So maybe that's why they're not seeing so many of those arrhythmias. And of course, with anything IPS derived, you have to make sure that there's no teratoma formation, no tumor growth from proliferation of certain IPS derived cell types. And they didn't see any tumor growth either on the heart, which is great, And I think that in itself would have been an amazing study to publish alone. But of course, like I said at the beginning, everything here is being oriented towards a clinical trial in people. And that clinical trial has been initiated, a phase 1/2 clinical trial in Göttingen, and I think Wolfram actually showed some of the results for this over the last couple of years. And so the very last part of this Nature paper was taking a heart from an individual who had received these EHMs prior to actually receiving a true human heart for transplant. Again, as the bridge, right? They had taken that heart that received the EHM and did a bunch of analyses on it, histology analyses, imaging analyses, and show that there was actually remuscularization by the AHM implantation in that patient, and so that heart was able to be stable for a number of months after the EHMs were, basically slapped on the top of the epicardium. So that's really exciting. It's an N of one. They just wrapped up the paper just by showing that N of one human heart that received the EHMs, and apparently there have been already over a dozen people who have received these EHMs in the clinical trial. So I'm stunned by this. I'm amazed by this. This has been a dream in the field for so long. It's answered a number of questions. It's not a long-term solution, like I'm saying. This is a bridge, adding these EHMs to somebody's heart to stabilize it before they can receive a real replacement. But nonetheless, it's another huge victory in the iPS cell therapy field.
Yeah, this is really impressive, and I was surprised by it because I mean I haven't followed the field as closely as you or heard the preliminary, so I was impressed. I know this isn't necessarily like a conceptual leap forward because there have been a lot of this has occurred to a lot of people, this idea of the band-aid. Um, but yeah, I was a bit shocked, and the how effective it was. Um, And especially in light of in recent weeks, I think just last week we heard about how the xenotransplantation, there was some kind of immunogenicity, it seemed, at a base level, that was a little concerning in terms of the prospects for that. We know about the arrhythmias and the long promised and great hope held out application of ESL. So this is great in a really pragmatic way. Again, as you've said multiple times, it's a bridge approach, and I think that is something that is really likely to reach the clinic soon on a broad scale, because it's not like this compassionate use application, right? It's like patients that you expect to live, tissue that you can recover, and you can really flesh out all the parameters. I think that there is a lot to be fleshed out. They were very transparent. I appreciate that more than anything. Um, in talking about how there was these osteochondral cells in like a third of the monkeys that were transplanted, they reasoned to that it was attributable to inefficient differentiation. So we're talking about off-target cell types, although there were no consequences to that that were measured or recorded. I think it's encouraging to me that they're looking for those things, and as you said, this is all geared towards human trials. So the scrutiny, you can imagine, is great, and the fact that they're including these in the academic paper shows that they're being completely transparent and comprehensive in their approach. And I think that gets to another kind of nuance detail here, is that while these are iPS cells, so the idea of like autologous is out there, or I wouldn't say implied, it's never stated or even implied. I think people have moved on from iPS. These aren't iPS autologous grafts. These are allografts and necessitate immune suppression, and that is kind of a hack in terms of the bridge approach, allowing for this ripcord where you can withdraw the immune suppression and the grafts will be rejected if there's any kind of off-target differentiation or complications. And because it's not central and grafted in the ventricle, perhaps blowing those cells up wouldn't lead to some acute fatal event. I don't know. Lots to be fleshed out, as I said, but a pragmatic approach, I think, that has safeguards is, I don't know about ready for prime time, but certainly ready for trial, and an exciting, much more, I think, practical approach to healing the heart, at least in the short term.
Exactly. That's the key point at the end that you made there, short term, right? I still think that the long term solution is going to be Xeno. The mass generation of immunocompatible xenotransplantable hearts from pigs. Um, but we are not there yet. We've talked about some of those studies ad nauseum here on the show, the clinical therapeutic strategies, and how those patients have been able to extend their lives for a couple months before this antibody mediated immune rejection, which I think is going to be a huge hurdle to get over. So, this is a cool parallel approach, and I think a nice parallel approach is not completely replacing the Xeno approach, but just as somebody in the cardiac field, as in the iPS cardiomyocyte field for 15 years, and who's seen the progression of this work from in vitro to in vivo, it's a really gratifying day, and we'll see how it goes for the rest of the clinical trial here.
Yeah, and I don't think anyone needs a reminder, but it's worth saying, right? There's a tremendous amount of effort that going toward this, and there's been, yeah, a lot of obstacles, many of which have been overcome, and many remain, some unknown. But the scope of disease is it's the biggest. It's the biggest. And it's growing. So it's justified, and every incremental step forward is major. And I do believe, like you, that the Xeno is going to provide a really, again, practical, pragmatic solution. But the number of hacks that you're going to need in order to get those cells to evade the immune system, which is a little bit of foreshadowing to the story you're going to come up with after me here. But let me move the show along here, talking about going to the other major application that we talk about of embryonic and pluripotent stem cells is the modeling of development, and something that is very near and dear to my heart and my bread and butter. No puns there. Uh, is pre-implantation development for the purpose of assisted reproduction. And the story I'm going to talk about in Cell is a resource toward those ends. And again, it goes back to the basics, but here in a way that's really applied clinically in the context of IVF, as I allude to, talking about cell identity and the cell transition, really. And while we talk a lot about the first differentiation event, right, between the trophectoderm and the ICM, there's actually a cell differentiation that precedes that, which is the transition from a germ cell, which is highly differentiated, specialized cell, to this totipotent zygote, where you have to completely erase all these germ cell specific epigenetic marks and get back to developmental ground state. And the second major transition, of course, between the trophectoderm and the inner cell mass, flanking those transitions is the zygotic genome activation. And what I didn't know before reading this, which I'm kind of embarrassed to say, which I should have, is that there's actually two waves of zygotic genome activation. The one that's familiar, at least to me and probably to most, where in mice at the 2-cell stage, at humans at the 8-cell stage. But there's an early wave in both mice and humans of zygotic genome activation on a much smaller scale. And in mice, that takes place just a few hours after fertilization, and in humans, it takes place in the late 1-cell stage. And the reason why this is relevant is because a lot of the failure of embryo development, both presumably in vivo, but it's been very clearly observed in vitro, is the failure of oocytes to be fertilized and reach the 8-cell stage, this point of the late wave zygotic genome activation. So understanding the dynamics of those transitions is really an important goal, right? And how do we understand them? Well, proteins, right, are very underappreciated because we're all focused on the transcriptome, the epigenome, because those things are measurable at small scale, and the technology hasn't really matured. I mean, we all appreciate proteins, right? Of course, those are the molecular machines. They do all the work. It's the business end. But I think we don't really have a handle on the dynamics and a really high resolution single cell, for example, means of measuring protein levels and activity in different isoforms and modifications. But the technology is improving, right? There's these mass spec based methods that have incorporated single cell technologies. There's a bunch of them, but they've all come out just in recent years. And they've gotten to the level where you can identify over 2,000 proteins from a single mammalian somatic cell without specialized equipment. So it's accessible to labs. And oocytes and early embryos are even more amenable to this, right? Because they're bigger. They have a lot more protein, and they are, in the case of embryos and single cell, zygotes, they're encased in the zona pellucida. So they're like encapsulated already, ready to be analyzed. And because of that, these ultrasensitive tech is able to measure like up to 8,000 proteins in humans and 6,000 proteins in mice, which is a little bit smaller. So this is the tool that was developed by a bunch of investigators led by Yidi Sun, Chen Li, Liangshan Mu, and Zhan Lu. Apologies for any mispronunciations there, all from the Chinese Academy of Sciences in Shanghai. And yeah, they developed this ultrasensitive proteomic technology they call CSUPT, and then applied it in a lot of mouse, but also in human embryos, pre-implantation stage, multiple stages. And as I said, they were able to scale up the number of proteins they were able to identify versus like a somatic cell by like multifold, fourfold. And they showed that there was, of course, I mean, this is a resource, so it was kind of academic in terms of the insight and application toward conceptual advance, but I thought it was still important. They showed that there were distinct proteomic dynamics between the mice and human, as you would expect. They identified a lot of regulators in the mouse in terms of discovery, and looked at those in the context of human embryos. And here's where I think it got interesting: is they actually showed in couples, in fertile couples, looking at more than 100 patient couples comparing good quality versus poor quality embryos. And they identified a distinct profile for poor quality embryos versus good quality. And it was key. There's a key point here. It wasn't influenced by the number of proteins detected, that signature. It wasn't like, predicated on how many proteins or what the age of the patient was, or how many cells were able to be analyzed, how many cells were in the embryos. So irrespective of all these parameters, kind of controlling, normalizing for those, the poor quality embryos were able to be distinguished by their signature, even multiple poor quality embryos within a single cohort from a patient couple. Um, so this is really important from like a diagnostic standpoint, but also looking downstream at specific regulators. They identified multiple genes, but we'll highlight one, this TPRX that they talked about a lot, TPRX1, which was a hallmark of good quality embryos. And on the flip side, they identified a marker, NCOA3, among others, that was a hallmark of poor quality embryos. So I think, from the diagnostic standpoint, I think this is really important if we're trying to understand or predict embryo quality, although at this point it's like destructive. I don't know that we can use the embryos for implantation or transfer. But still, of course, the technology should and will progress. But also mechanism, just trying to understand why and how uh, these embryos fail to reach the 8-cell stage. And the early data here, looking at some of the genes that were identified, suggests that yes, it is a failure to adequately fire up zygotic genome activation. And some of these factors, if they're identified, the ones that are lacking or the ones that are present, may be targetable to improve the efficiency of pre-implantation development in the clinic. So for me, very exciting. And also, I think for all of us, a tool that's going to provide higher resolution proteomic analysis.
Yeah, really exciting study, an amazing data set, again, helping to open that black box development. The great part of this study is that they use both mouse and human tissues for these early analysis. I mean, that is one of the limitations, that the human data set wasn't huge, understandably. I mean, there's ethical limitations in terms of how far you can actually go to obtain some of these samples. So they mentioned it. A lot of the human mouse embryos lack samples from the morula, trifectum cells from the blastocyst stage, and of course, much later on too. But an amazing reflection on the power of single cell proteomics. We have a few folks here at Cedars-Sinai, where I'm at, who are really world leaders in proteomics, single cell proteomics. Jenny Van Eyck comes to mind, and that some of these new technologies, mass spec, that enable the proteomic analyses are allowing us to shift things perhaps in terms of our omics analyses more on the protein side of things, because it's the proteins that are really doing the work in the cell, right? We're so stuck in thinking of transcriptomics, in part because it's so accessible. Everybody can do bulk RNA-seq, and everybody can do single cell RNA-seq now, but not everybody has access to these high-powered proteomic analyses and technologies. But perhaps that bar is coming down, and within the next 10 years, proteomics is going to be replacing transcriptomics in terms of the analyses and the bulk workhorse analysis that you would do at the omics level. I don't know. That's a dream, and this is a paper that's pushing us towards that dream. Really great study. Limitations aside, I think just a reflection of the power and the advancements in single cell proteomics technologies.
Yeah, and I mean the tech marches forward, and you got to look at everything over again. But I think another, kind of incidentally, maybe not on purpose, a group of people that will be interested in this. Not that this is what the group was going after, was like benchmarking for the human embryo models. I mean, it's a different system lacking the zona and et cetera. But I think that when, you know, and granted, this is pre-implantation, but we're talking about the blastoids, right? So I think that when you think about why the embryos don't look right or fail to really recapitulate some developmental events, I think we need these kind of benchmarks, and transcriptomic has been really well established. But these proteomic resources, I think, are going to be useful across the board for all of us stem cell scientists.
All the omics. Let's do all the omics. But of course, the key to doing all the omics is to bring down the costs for all the
Yes.
omics. And transcriptomics has definitely gotten there first in terms of making it more accessible. And proteomics is, I'm sure, right down the pipeline, right? So moving on to a cell stem cell paper, which is also in the realm of development. This is a pretty exciting study looking at bipaternal offspring, adult bipaternal offspring that are generated through direct modification of imprinted genes in mammals. And this is a one of those studies that caught the public's attention, the public's eye, because of its implications. Bipaternal, two male parents, quote unquote, to generate embryos that can last into adulthood. And this is with the caveat that this is a very inefficient process, and what you alluded to in the beginning, this is sort of a hack. I mean, it is a hack. There was a lot of genome editing that had to happen for this to actually work. 20 plus knockouts and various different genes to actually make viable pups with what they actually showed increased cloning efficiency. But the other part of it is using this quote hack approach of modifying the imprinted genes. They also made it's quote enhanced diploid embryonic stem cell lines with enhanced pluripotency. This kind of like a side project that contributed to the main project. But let's get into it. So imprinting abnormalities have a huge challenge, and we've talked about this on the show, and I think you've actually covered some of these studies. There's a huge challenge with these imprinting abnormalities when it comes to embryonic stem cells, right? iPSCs as well, animal cloning. This is a huge barrier to overcome in imprinting to really make these bona fide IPS lines, improve IPS lines, improve cloning, improving all sorts of downstream reproductive applications. And there's not a universal correction method that- to address this, but they're going to take a brute force approach in this particular study. So they targeted these imprinting abnormalities at their source, at the embryo level from same-sex parents. They just CRISPRed the heck and modified the heck out of these early embryos, aiming to establish a stable, maintainable imprinting pattern de novo in mammalian cells. So using bipaternal mouse embryos, which actually exhibit severe imprinting defects and are usually non-viable, as you might expect from two male mice, giving rise to an embryo. They had introduced they the whole gamut here of mutations, frame shift mutations, gene deletions, and I'm not going to go into naming all the modifications that they made because there's 20 of them. Regulatory edits at 20 key imprinted loci. And after they did all this hacking, they ultimately were able to achieve the development of fully adult mammals from these bipaternal mouse embryos, but with a very low survival rate. Not surprising. This is a hack to get 20 genes to go this far, and I'm sure there's more that you have to modify to make this a more viable solution and improve the survival rate here. But still, regardless, a really impressive study. It shows strong evidence that imprinting abnormalities are a primary barrier to unisexual reproduction in mammals. And this could also potentially improve developmental outcomes, importantly for embryonic stem cells, improve pluripotency in these populations, maybe better embryonic stem cells overall, and maybe even improving the efficiency of cloning in animals. And their words, opening new avenues for advances in regenerative medicine. Sure, it's a broad envelope, a broad umbrella, um, but this is a critical hurdle to overcome in these imprinting abnormalities. Um, and I think this is an exciting application of CRISPR genome editing to really just hammer and nail your way to these viable offspring from these bipaternal mouse embryos. Again, very, very low efficiency, but you got to start somewhere, right? And we'll see how it goes from those.
Hack it up. Hack, hack, hack. I mean, the story here, I think I haven't really looked at the news lately. I've been pretty busy. But I could imagine this might have been hijacked because of I mean, it is in the title, first word by paternal offspring, right? So not the first, adult by paternal offspring, maybe even more sexy. But the story to me, this isn't a story about unisexual reproduction. It's a story, as you just said there at the end, about CRISPR and about the amazing ends you can meet by just marching along. And I don't think we can understate how crazy not clinical this is, like impossible and unsafe. And like from the bones of it, it starts with a haploid ES cell. I mean, we're talking about so many interventions, even more than the last time where they had to get the Y dropout and so and what I'm saying there, I'm not throwing shade here. I don't think the authors were out here talking about uh, reproduction in clinical context. I think that they, like most scientists, I hope, appreciated this was about the basic science and understanding of imprinting and how these single modifications can very specifically I don't want to call it abrogate, but address or kind of like isolate these phenotypic uh, problems of failures to develop. But like you, I mean, you said it. If it takes 7, they get overgrown, non-viable fetus. Then they get up to 15, reduced body organ size. Okay, they go 18, viable pups. Yay, 20 full placenta at 20, and then they stop there, right? And they publish. But I don't think anyone in that group is like, we did it. I would assume that number one, all those modifications, as they noted in the limitations, may have unintended consequences to development and or homeostasis and the lifespan of the animal. But also, like, is it enough? I don't know. Why is it going from 19 to 20? That's it, that one thing? I don't think so. So as you said, there's probably going to be a few more things to get the efficiency up and to address some like downstream pathology that's undermining the success in the first place. But again, back to this triumph and intuition. I mean, I'm really impressed. How do you come up? How do you go from 7 and be like, okay, you know what? Let's try this. I guarantee you it wasn't like they hit it right on the head first time. So it was great intuition and a lot of hard work. I mean, I don't know how many, but 100 maybe they had to try and screen. This isn't like a screening approach. They had to see if these will generate viable embryos. It's a crazy amount of work. So I'm very impressed and annoyed that people are trying to make this a story about clinical application when it should be a story about like the basic science and imprinting and insight gained there.
Yeah, and a story about the power of the tools, like you're alluding to. We've covered some high profile papers so far on this roundup, and they're all kind of unified by the fact that they're enabled by this new generation of tools that we have. The first study I talked about was the engineered heart muscles. That's because of IPS, right? That's the reason why that emerged and perfecting the differentiation approaches. I mean, the second story we talked about was on single cell proteomics. That's what enabled you to open up that black box of embryo development. And then this is a story about CRISPR and the applications of CRISPR. And certainly there have been in vivo clinical applications of CRISPR, and this is not anywhere close to prime time, like what you're alluding to. CRISPR, as we know, has so many off target effects. And how do you validate those and identify those and correct them and really make sure that the modifications that you make are clean? And you're totally right. I doubt they're stopping at 20, and I'm sure they're going to brute force their way to beyond that. But with every modification you make, perhaps there's an unintended consequence, especially if you're using CRISPR. So maybe this is another reflection on how some of these next generation genome editing technologies like prime editing and so on might be the way to go forward with with clinical applications, I don't know, maybe since they are a little bit cleaner than your old school Cas9. It's crazy that I'm calling Cas9 applications old school now. It's only been about a decade since we first started using Cas9 in vitro and in vivo, but it just tells you how quickly this field is moving.
Yeah, it's like Cast Nine and Stone Tools in the catalog. Um, but you said it, not clinical. I said it, not clinical. Who cares? It's amazing conceptual advance, and sometimes you just need to demo the possibility, and this is it. This is live-born pups viable. And as we've said before, get your boy Seabass on the line. We gotta try this out for species conservation. Maybe that's the first move, and that's something that's a lot more palatable to me. But you talked about the tools, and here I'm talking about in our last roundup story. It's not even a stem cell story, kind of at all, but it's going to be pretty soon. I think we're going to see this tool applied a lot in stem cell systems. It's an inducible protein tool, and inducible proteins. We're probably most familiar with them in the context of optogenetics, right? Where you can trigger this protein conformational changes with light. Probably going to win the Nobel Prize. And the great advance there is that you have tremendous precision. In time, obviously, it's inducible, but in space, you can focus the light. But in 3D systems, it's a little bit tougher, right? Because of light scatter. And inducible proteins like optogenetically regulated proteins would be a boon for the commercialization of stem cells, which is why I'm kind of covering this story. Just think like, you have cells that are in a patient that you want to, regulate inducibly. You could put a heat or a cold pack there, right? Because with optical control, there's the scatter. You can only get to about millimeters depth of human tissue, right? But temperature, you can get down to centimeter length scales, either with like a heater or cold pack. And if you want to talk about precision, even you can use focused ultrasound that you can get sub-millimeter scale spatial resolution of heating tissue. So you have all the benefits of optogenetics, presumably, putatively, but also have this scalable thing, which again, think about it like in a patient, if it's regulated, or like in a bioreactor that's really cloudy. Maybe the light won't all the way get through because of the opacity. The heat would be a good way, or even like interfacing. And this is the cool bit of a mind f thing for me, is you have this tool that interfaces with the organism's own stimuli, right? So you have a system in there and engineered proteins that can detect intrinsic stimuli, physiological temperature cues like fever, like inflammation, to activate a response. So a lot of really cool applications. But I'm sure a lot of people have thought of this and they just haven't had the right tool. There's just not a lot of proteins that have temperature sensing modules. But there are a few, and this group, which was led by Lukas Bugaj at UPenn, they created this whole catalog of thermoresponsive proteins that they named MELT, and that name is money. Going to be widely used, even though it's a kind of a loose acronym. Stands for membrane localization using temperature, and it's a modification of this naturally light and temperature sensitive protein called BCLOV4. And they made, as I said, a catalog of these proteins. And naturally, the protein, it forms this oligomer and translocates to the plasma membrane when temperature is lowered. And they have this switching temperature, what they are able to vary with this library that they generated. So they have switching temperatures ranging from 30 to 40 degrees Celsius. That's a pretty useful band. And they have two of those that operate, one at 37 degrees and one above 37 degrees. So like I was alluding to there with that whole like fever model, they show that this thing was really modular, that they were able to control using heat, all kinds of signaling and proteolysis, nuclear shuttling, cytoskeletal arrangements and cell death. And that's where they reach the kind of level of impact and relevance by showing an actual in vivo system that they could thermally control the cell death, apoptosis, I think, of human cancer cells in a mouse model. So right there, I think, generating the tool, great, but applying it and really giving a vivid example of how it can be applied. And across all these different or cellular compartments, combining two different things to make a nuclear localization of protein or cellular localization of protein. So effectively, you can do whatever you want in response to this switching temperature. So a really cool tool that I can't wait to see used in ES, inducible differentiation based systems. The Nature Methods paper. I'm sure the plasmids are on Agene. Get on there and get going. Arun, get it and clone it and then send it to me when you got it working, will you?
Will do. I love these stories. It's such a cool reflection on the power of engineering, right? Synthetic biology is all the rage these days. And we've talked recently on the show about folks at UCSF who have been doing some of these synthetic bio approaches. This is, of course, a group at UPenn. But you're right. This is, I think, very, very powerful because temperature is something that the body is so in tune with. And it's something we don't think about a lot here on the show. Life and human life is compatible at a temperature range that is like within 10 degrees Celsius. Like standard body temperature is what, 98.6. You go up to 102 degrees Celsius, you got a fever and you're pretty sick. You go down to 95 degrees Celsius, you're probably dead. So we live within such a tight range of temperature, and being able to fine tune protein activity based on a degree Celsius here or there, that's an incredible accomplishment and not something that should be understated. Just how powerful this is, because you're right. I mean, that's the dream, is being able to tune these melt variants and tune these synthetic proteins to endogenous body temperature. And maybe in the realm of science fiction, we can think about a situation where everybody has these melt proteins coupled to, say, the immune population in your body, and perhaps depending on a fever or a slight alteration in body temperature, these melt proteins are going to activate and selectively target regions of inflammation to rapidly reduce that sickness or that illness, infection, whatever you want to call it. I think a million applications here. I think the cancer one was also really cool that they alluded to here in the show, this programmable cell death. So I'm a big fan of engineering. We've talked about basically 4 studies here that are utilizing tools and applied tools in stem cell biology. And undoubtedly, this is going to be a really exciting tool for the toolkit in stem cell biology, but also just across cell biology in general.
Yes, this has been a roundup focused on tools, and we're going to make it an episode that highlights tools and toolkits featuring two of the greatest tool makers in our field. This is a cool pair, and we're going to get to talking with them about how they've set us all up with their toolkit. But first, I got a tool from STEMCELL Technologies for you, and that's the colony forming unit assay that can be used as a potency test for hematopoietic cell therapy products. Learn how to identify and count colonies with this free on-demand course on the human hematopoietic CFU assay. To learn more and register, visit www.stemcell.com/human-CFU course. All right, everybody. Today on the show, we have what is a very special treat for me. As many of you know, I love developmental hemogenesis, but I'm pretty sure it's going to be a real treat for all of you as well. We have a 2-for-1 here on the show today, although talking very much about a core element that is shared. We have doctors Andrew Elefanty and Elizabeth Ng, who are the principal research fellows and co-group leaders of the Blood Diseases Laboratory at Murdoch Children's Research Institute. Dr. Elefanty's research uses human pluripotent stem cells to study the development of blood cells and diseases of the blood system. Mimicking the diseases in the lab allows testing of new combinations of drugs that could more effectively treat leukemias or stimulate blood growth in children with bone marrow failure. His lab also focuses on investigating whether tailored blood stem cells could be made from the patient's own cells for transplantation. I mean, this is the holy grail of stem cell science. Because that would prevent graft versus host disease, which we all know is a scourge. Also, Dr. Ng's work has identified that the expression of the HOX-A genes during differentiation is critical for generating hematopoietic cells with a transcriptional profile similar to the pre-HSC seen in the aortic gonad mesonephros region of the human embryo. Her current work focuses on the challenge of maturing these cells further to produce engraftable HSCs and to develop in vitro niches that mimic the bone marrow in order to sustain them together and with their collaborators, of course, which is a long list and what might be called the crowning achievement of decades of work, not just in their labs, but all over the world. Doctors Ng and Elefanty have made a really a broad swath of important contributions the generation of genetically modified human stem cell lines. Today, we're really going to talk about the blood, as I've mentioned many, many times, but we're going to be all over the place. Doctors, thank you so much for joining us on the show today.
My pleasure.
Yes, you're welcome.
Yeah, thanks so much for being here. And like Dale is saying, the timing is really great given the amazing study that your lab published recently in Nature Biotech describing long-term engraftable hematopoietic stem cells differentiated from human pluripotent stem cells. I mean, we've often described the generation of bona fide human HSCs from pluripotent stem cells as a holy grail remaining in the stem cell field. Folks have been working on it for so long. So it's beyond exciting to see a study like this come out. We covered it on the show a while back. It really gets us one step closer to that ultimate goal, like Dalen was alluding to. So we'll talk about the paper in a little bit. We'll devote a lot of the show to it. But first, to help our listeners get up to speed, perhaps you could both tell us a little bit more about your research interests in your own words. So what is the lab focused on right now?
So Andrew and I work very closely together, and we have for many, many years. And in fact, the way we approach our research is more as a pool. So even though our research interests can be framed as either disease modeling or developmental biology, the tools and techniques and the development of the protocols and the research questions we do as a whole group, because you can imagine that one thing informs the other.
And I guess the only other comment I made relating to your lead-in about the collaboration is to mention that we've been very fortunate that over really the decades that it's taken for this type of research to evolve has been driven really not only by Elizabeth and myself, but also by a close colleague, Ed Stanley, who we've worked with for over 25 years. And I think that in there is really part of the I guess the secret of success, or the reason that we've been successful, is we've had a stability of really sort of a long-term collaboration between scientists who've got sort of complementary skills and complementary sort of expertise, and that's really enabled the work to sort of flourish. And I guess also returning to the point of your question, a lot of the principles underlying the way we've approached the research has really been to try to investigate, to interrogate how what happens in the embryo can be adapted to what happens in vitro in addition. Maybe Elizabeth can sort of chat a little bit more about that.
So the way I would frame it is that, if you like, many years ago when we were all younger and our groups were smaller, Ed, Andrew and I began working together, and we did bring complementary skills to the table in terms of our skills in either tissue culture, protocol development, or molecular biology. And the three of us together over the years, we've become three different groups, or we have three different focuses now. But we could only do it by working together as a team for more than 20 years now, isn't it?
Yep.
Yeah, I mean, like no one else, I think I would identify that triumvirate. I mean, it's hard to call it triumvirate because that has a connotation of like dominating leadership. Whereas I think you guys have been so collaborative. When I hear those names, Ng, Stanley, and Elefanty, that's what I think. It's not a single group leader. It's a whole bunch of tools and insights into development across the board that have been generated that have all those names attached. And I don't think there's a group like that is truly collaborative across the board. And as I've said, I mean, this show, we're going to talk about hematopoiesis because that's the latest splash, and you could argue maybe the biggest. But like you guys have had a broad impact across mesodermal, endodermal, ectodermal lineages even. I mean, indeed, there's all those different reporter lines you've generated. I'm sure Arun has used one. I myself use that NKX 2.5 knock-in that you generated. Also the mouse. No one talks about that in Harvey Lab. I use that mouse for a paper that came out a few years back. Literally, we're talking hundreds, if not thousands, of papers that have incorporated this and other line, the MixL1, insulin, all these other lines. And I can see the link, maybe how they circle back to blood, but many, like, let's take, for example, NKX 2.1 for identifying basal forebrain. I mean, I don't see the link there at all. So I have to ask, what drives those forays into the, you have your expertise going in, your knowledge, you have your goal, the blood, I think. So what drives these forays into these other lineage-specific derivatives? Is there a link there that I'm not seeing back to blood? Or, I mean, it could just be your excellent reputation where you just have collaborators and banging down your door saying, let's make this line, or is it just curiosity, one thing leads to another? Tell us, how do you get so far afield?
If we look back into the past, our philosophy of how we were going to do anything was to be guided by the embryo. So the three of us have a strong background in developmental biology. And the way we approached some of these problems was essentially to ask someone who does it well. And the embryo is the one that does it well. So what we were trying to do was to take lessons from what we knew occurred during embryonic development, and that was first in the mouse, and we then transferred to the human. And we tried then to model those things in vitro. And the best way to model them was to take what we knew and to try and objectively have some form of objective measurement for the timing of events and for when certain things had to happen and for combination of when certain important switches and things had to happen. So the best way of doing that was to generate reporter cell lines, because then you could objectively measure what you were doing in vitro.
And to expand on that, and partly to circle back to the question that you asked about the specific reporters, one of the early sort of conceptual sort of thoughts that we had is that you can break up the developmental process to blood or to anything else as a series of stepwise decisions. Now, I mean, it's not exactly like that. Its development is far more of a continuum, but pragmatically, it was easier to do this. And if you look back over the reporters that we generated over the years, the earliest ones were to help us with lineage allocation. In other words, in trying to help us decide what cells were turning on genes which were relevant to mesoderm, and we knew that things like blood and endoderm came from mesoderm, so hence the initial interest in MixL1. Then we also had interests which have continued in parallel into certain endodermal lineages, and in particular, Ed's had a strong ongoing interest in development of pancreatic beta cells, the cells that make insulin. And as part of the suite of genes that regulate pancreatic endoderm formation, hidden amongst that lot, as well as obviously insulin and PDX, is NKX 2.1. Now, it just happens that NKX 2.1 is a bit more promiscuous and is expressed in tissues like the lung and also in the ventral forebrain. And it it happens that to develop the cultures in vitro to the stage where the reporter is expressed, it's easier to do forebrain and lung. And so we use those initially as tools or readouts to determine if the reporter cell lines were faithfully reporting, the gene expression that they should. And that's why we ended up getting sort of sidetracked and fascinated with the ventral forebrain. And, interestingly, that cell line has been very widely distributed, very widely used, and, is a really nice reagent. And that's something we've been very keen to do. That by that I mean to distribute our lines really to any research groups that ask for them, and we've been rewarded by two things. One, by the fact that people have been very generous and included us in their research and stuff in areas which are not really primarily what we do. But secondly, and more importantly, it served to validate the tools that we made in independent hands, and that's a important thing.
That's a very important thing, and the same thing with
and relevant for the protocols that Elizabeth
that's right. The same thing with the methods that we develop in house. So, I suppose one of our philosophies as a laboratory or as a large group is that we do as much as we can try and share our reagents and our technologies with either particular collaborators or with people who are working in different areas, as Andrew says, because it helps to validate the tools and technologies that we develop. But the other thing is, I suppose, a philosophical one, and that is that we're totally aware that no one lab can do everything, and no one lab can answer all the questions. And for many of the questions that we're asking as a field, you need more than one brain, more than one group, more than one outlook in order to solve them, to create a more complete picture. So in that sense, it's been our philosophy, I suppose, to share as much as we can.
And in fact, that actually has stood us in very good stead, because we've also found the opposite, that people are willing to share their data and their insights with us as well, and so that it works both ways.
That's
if
right.
you're if you're able to be generous with your reagents and your advice, you find that often you learn things which otherwise you wouldn't.
That you never would have learned before. Yes.
Yeah, absolutely. And, I can speak from experience in saying those lines have been such a tremendous resource for the community, but also for myself as a cardiac biologist. I've used that NKX 2.5 reporter throughout my training. It's also kind of inspired me in my lab to generate my own reporter lines. It's I think with CRISPR and everything, it's easier than ever to do these kind of things. And, seeing is believing. It's really where I think a lot of us are visual, right? Where we love the visual, we love the seeing of these different lineages, and it's just a tremendous tool in that way. Like you said, the embryo does it well, but actually I think the embryo does it best. And as in vitro biologists, we're just trying to approximate some of those embryologic principles, developmental timelines, not always getting it right, but we're doing the best we can, I suppose. So, we're talking about basic developmental biology for this in this conversation so far. Of course, the work that you've been doing recently has a lot of translational implications, and in particular, the work you've been doing in the HSC generation from pluripotent stem cells. I mean, like I mentioned, it's been described as a holy grail in the stem cell field for decades now, in part because of its incredible clinical potential. So let's actually jump ahead for a second from basic to clinical translational. A study like the Nature Biotech study ultimately gets us, closer to that goal for clinical translation of stem cell derived HSCs. But Could you actually tell us what's missing before we get to that goal of translatable or transplantable, clinically applicable HSCs derived from pluripotent stem cells? How far are we from actually meeting that goal?
Probably, I would say that the study that we've just published gives us great hope that we will be able to translate the research into the clinic. One of the bridges, if you like, that we still have to cross is we need to expand the research. We have to be able to do what we've done in-house, but we have to expand it. We have to do it with many, many more cell lines. We have to, if you like, shore up the protocol is robust, but we need to, I suppose, continue as much as we can to improve it. And the other issues that we need to face in order to translate anything into the clinic are often practical ones, which scientists do not necessarily, well, basic research scientists, which is what Andrew and I are to consider things that normally we would not have to consider. And that is about manufacturing, about scale up, about patients,
And also, too, there's the practical considerations of ensuring the safety of the
Correct.
product as well. So they're things which are not quite so paramount, in the research application, but become really, front and center when you're looking at the translation. So, as Elizabeth says, the protocol has to be, better, faster, bigger. But also you have to have, ideas in place of how to ensure that it's safe.
Safety, yeah.
And I suppose the other point, which was sort of implied in your introduction, but we haven't sort of formally said, is that one of our real goals is to try to explore this as an autologous therapy rather
Correct.
than an allogeneic, and we can go into that sort of further if you wish. But that's underlying, the comments that we made as well.
Yeah, I mean, we can dig into that because that, I think, is the key in terms of generating a product from a patient, as you guys, I'm sure, have thought about and spoken to industry people about, is that adds another hurdle, right? When it has to be personalized and you got to run through that protocol every time. Every patient is a IND, right? As they say. So it's a high bar. And I mean, let's be honest, like you've already surmounted an obscenely high bar. This is something that people have been trying to do from the beginning. And I, as someone who has failed with hundreds, I mean, over 500 mice that I've transplanted in my, I went at it for probably 4 or 5 years. And that's after a long diff and, sorting cells. Like, this is a ton of work to do this. And a lot of people have taken a swing. And I look at what you've done, and I would just want to dig in, get a little granular. And I'm sure you guys have been through the ringer. I mean, given the extraordinary nature of this claim, but like oddly, this was only about 15 months in Nature Biotech, which is bizarrely like a short review there. So I imagine it must have arrived at the editor's desk as a pretty mature story. So you guys have been working on this for a long time. And how big a delta it is from what people have been able to do before. So I'm sure you've gotten this question before, like, what about like the phenotype? When you talk about the cell, the engraftable cell, you have the CD34, which is a pretty broad descriptor. And of course, it's a subset of those. But I mean, for me, there's so much more in there, right? As a developmental hematologist or someone with an interest, like the HOX-A patterning was big. There was the defined media that is a big and critical, I think, to getting a robust and reproducible product. But it still leaves me wondering, despite how repeatable, robust, what's that cell of origin? But then it occurs to me that maybe you do have to inject the whole aggregate, right? Because maybe you need not just the engrafting cell, but you need some niche cell or some kind of adjuvant cell. There's so many things. There's been failure for so many years that I imagine that there's a multiplicity of influences that need to converge on success here that you guys have reached. So there's so many questions, maybe not like clear answers. I don't want you guys to, you could probably go on for hours, but if you could just in a nutshell, like, why do you think this works so well? Why is it so robust? What is the key difference?
I would say that the importance or the key difference lies in the, again, a mixture of science and a mixture of practicality. So the science is the developmental biology. So this was a long time in the making. Probably the biggest the underlying or the underpinning breakthrough to this was the Hoxsey paper, because that really told us not only did we know where the true hematopoietic stem cells came from, and this is obviously work that has been done by other luminaries in the field, looking at human developmental hematopoiesis. So we know about the AGM being the site of where the first stem cells arise from during development. But being able to model that in vitro, there were several things we had to overcome. And the main one was the realization that if you make blood in a dish, all the blood looks the same. It expresses cohorts of similar genes. It looks there's nothing to tell it apart. Understanding those different waves of hematopoiesis during development, and then being able to figure out what was different between them, and being able to model that in vitro, that was the underpinning bit of data. So realizing that one of the main differences between the yolk sac and the AGM was the expression of genes of the Hoxsey cluster. So that straight away told us if we wanted to make the right precursor mesoderm to give rise to the right cells, we had to pattern it correctly right from the beginning. So what you do on day 1 affects what comes out, at day 14. And you can't go back. If you don't get that right at the beginning, what comes out will never work. And we know that because we proved that many times.
And in fact, there's further than that. I think one of the other answers to your questions is it's not just one thing. There's
Correct.
about 4 or 5 points along the way,
which
perhaps you can comment on that help to explain, what the answer to your question.
So Andrew has this saying that drives the lab absolutely bananas. He says everything is important.
Yep.
So at every stage of the differentiation, we had to compare what we were doing with what we knew of human development. Now, the other really big important thing here too is that at the time we were doing this, we had no real roadmap, because you must remember we were looking at early stages of human development, which of course were not accessible to us as scientists. Yes, we had mouse models and et cetera, but in more recent years, what we've been able to do is we've been able to work with researchers who have been able to look at human material, at human embryonic material, and provide us with important signposts, as it were, so we could compare what we were doing in vitro with what was actually happening in the human embryo. And by being able to do that, we could look at where we needed to improve either the differentiation or what we were not understanding about the tissues that we were making in vitro. And that actually was really a crucial thing that we had to do.
So specifically, really, it's the confluence of not just the protocols that we're developing, but the ability to have that interrogation of human transcriptional profiling of embryos and functional studies of embryos. So that's really the work that was started by Alexander Medvinsky in his seminal paper of 2011, and in the papers by Hanna Mikkola, over the last 3 or 4 years, which have, been sort of the source of very prominent publications. And we've been fortunate in particular to have an ongoing collaboration with Hanna really since, 2015, 2016. And that has enabled us to have, access to her insights and her information. And so really part of the collaborative and sort of, group nature of this research, involves, and includes the contributions of these sort of, other scientists who've provided, necessary and complementary information, which as Elizabeth alludes to, gives us the benchmark. And so in that sense, also consistent with the sort of model that we've had over the years of development being a stepwise process, that gave us the first step as the patterning. And, to use a corny analogy, if you're in New York and you want to go to LA, if you don't start off, heading west, you're not going to get there. So, that's really the practical way of thinking that early patterning, makes a difference. And then along the way, I think, particularly in this protocol, there's sort of a couple of other sort of key elements in that we talk about. And perhaps you can sort of elaborate a little bit on those other sort of bits in it, which all add up, I think, to get something which has a chance of success.
So every protocol or every evolution of any protocol is an iterative thing. So you realize once you start on the experiments or the development of these, that as you go, you're going to need to modify things. So I think it's again, that's part of our philosophy as a group, is we're not afraid of change. So you will see if you read the paper that the protocol has gone through 3 evolutions, as it were, and that was necessary. The first question we asked, okay, we know we need to pattern intraembryonic mesoderm, and we can detect that using HOXA gene expression. But the other thing that we know from ourselves and from the literature is there are many ways to make mesoderm. So, and we know the signaling molecules that and growth factors that are involved in that. So how do we choose the right ones? Or what combination, or is there a right combination? And so the first question, if you like, which was part of protocol 1, was choosing the right mesoderm. And that was why, if you look in the paper, there were 12 mesoderm induction conditions that we used. And again, this is from work that's come from other people's labs, including Gordon Keller's lab, Chris Sturgeon, Andrea Dettardi, Igor Slupkun, people like this, who have also published in this area. We knew some of the molecules to use and to look for. And we also knew that you needed, for instance, retinoid signaling was involved in all of this. So we knew what the overall cocktail was to make the right mesoderm. Then we had to actually assay it. And so that was, if you like, the first step in the protocol. The second one, if you like, was once we had chosen the conditions to make the right mesoderm, we then had to look at the rest of the signaling cascade, the duration of signaling, in particular for the retinoid signaling, because it's present at that time in the embryo during development. So that told us the second piece of the puzzle. But the third piece of the puzzle, which came later, was in fact looking at the correct patterning of the hemogenic endothelium and where it came from. So now we go back to the embryo and we look at the formation of the artery, the dorsal aorta, and the hemogenic endothelium in the human embryo. And what that told us is maybe one of the reasons that at that stage our protocol worked but was inefficient was we were not patterning the hemogenic endothelium correctly. And that led us to the third stage of the protocol, which was actually modulating the VEGF signaling. And so as you can see, you can't do all that in one hit. You need to actually walk through the steps of the things that you're actually making in vitro and set them down. So and test them as you go along. And that's, I think, part of the reason for the paper seems complicated, but in actual fact, it's walking you through the journey that took us from the beginning of patterning the mesoderm right through to making the right hemogenic endothelium, which would give rise to the right cell.
And in fact, I think that doing it that way is actually is informative, because it does allow the reader to at least understand what the evolution of the thought processes, was. And as Elizabeth alluded to, it was really, pulling together bits of information that we had, found out ourselves, or that had been found out by, other key laboratories who've worked in this, worked in this area. And, we were, very fortunate in the, in understanding the VEGF signaling to have an ongoing collaboration with Professor Connie Bonifer, who was in the UK at the time, who's worked on, understanding the gene regulatory networks associated with signaling, for many, many years. And, and had recently sort of published that that VEGF signaling, impacted on the expression of RUNX1, which, we had also found was, one of the really key regulators of the hemogenic endothelium. So it's essentially, it's a confluence of, information and ideas, gathered from, a lot of, scientists who've been, working in this area for a long time. And that's what sort of put together this series of, of steps. And so in a way, the eventual answer to the question you asked a few minutes ago was that there's about 4 or 5 steps along the protocol where what we've done is been just slightly different from what's been done before, and that's why it has started to work.
Yeah, modern science is just so exceptionally collaborative. I mean, this is a cross-world collaborative effort. You mentioned Hanna Mikola, who's just down the road from me at UCLA, but of course your ongoing collaboration with Ed Stanley, many other folks who have contributed to making this work a reality from around the world. You're of course at Murdoch Children's Research Institute, which has become a powerhouse in translational applications of pluripotent stem cell biology, in a place that uniquely enables this sort of work to happen. And in fact, a good friend of mine who I always end up hanging out with at the ISSCR annual meeting is none other than Dave Elliott, who was the one who made that NKX 2.5 reporter line in the first place. And yeah, I just, for some reason, I always end up hanging out with him at ISSCR. He's a hilarious guy. He's a really
the only one that says that.
I hate to say it, but Dave is not very choosy.
Fair enough. That's fair enough. Yeah, no, he's a riot. He's an interesting guy. And he's a group leader in the heart disease group. He focuses on in vitro modeling of drug toxicity, cardiotoxicity, which is what my lab focuses on as well. A lot of overlap there. I mean, in general, the MCRI has this amazing research program, a worldwide reputation, in large part thanks to the work that you've done. Could you tell us more about MCRI, what makes it such a special place, in particular for stem cell biology?
Well, I guess that one of the things where, in a sense, we've been fortunate in that Ed, Elizabeth, I, and Dave, because at that stage Dave was still a postdoc working in our lab back in about 2011, 2012, were recruited to the new Children's Hospital in Melbourne, where the Murdoch Research Institute was sort of situated. And really, we came there with the promise of what human pluripotent stem cells could bring. And, we really had the idea that, well, we could build a focus of human pluripotent stem cell research here, because that was sort of something which was clearly, lacking in the Southern Hemisphere and in Australia. And I guess we were fortunate that after we moved there, the Institute was able to recruit, some really, big scientific names and scientific players in this sort of space. No larger than Melissa Little, who as the chief scientific officer there, heads the kidney group and is the CEO of the Renew International Consortium. Enzo Perello, who with Dave also heads the cardiac group. We also have key scientists recruited in neurosciences with Sylvia Velasco. We've got newer, younger scientists recruited now, who do muscle research, like Richard Mills, Shireen Lamonde, and John Bateman, who do cartilage research. So we've been able to be very fortunate that the nidus that we started has enabled the expansion of these groups, and that having the critical mass there, is arguably really one of the strongest areas of human pluripotent differentiation in the world. And look, that's been incredibly exciting to be part of for the last sort of 10 to 15 years. And I think that's, from that point of view, regenerative medicine, being, closely aligned with an institute which has got strong interests in genomics and is, integrated into the largest tertiary children's hospital, in Australia, is really a unique, combination of features. And obviously the other thing is that this has been strongly supported by our director, Catherine North. And I think that you need support from the top for these things to work. And I think that's been our good fortune to be part of that.
The other part, I suppose, of the policy of our leadership at the Institute is, in fact, that us as an institute, we work closely with the Children's Hospital. So for us, Andrew is a physician, but for someone like me or Ed, who's a scientist, what the Institute has helped support is the interaction of scientists with the physicians at the hospital. And this has actually been hugely important in helping to frame how we think about some of the problems or the possible translational aspects of the work that we're actually doing. And being able to also speak to patients and patient advocates also has something that we've been able to do at our institute. This, I think, has been very, very important in giving people like me, for instance, a much broader view of where our work and our research fits into the larger global picture.
Yeah, I mean, the Institute is amazing. I think one of a kind, as you describe it. I'm, being educated as to how actually exceptional it is, and how it kind of blazed a trail in terms of establishing that interface in a clinical space, creating these collaborative working groups, great leadership, as you say. But I think you guys, your group, the three of you in particular, I think deserve a ton of credit, as you described, creating the nidus. But I would say that nidus suggests like a tiny little crystalline entity that seeds something. Yeah, it's that. But you guys were also the anchor. And not only, I think, in terms of the strength of the science and the tool set, but I think conceptually, as you're describing it, I really feel that you guys took the lead with that idea. Of course, everyone's aware of it, but you went out there and created the tools to show it, that you have to model the embryo. The embryo is the best teacher, and these reporters are an undeniable indication of realization of cell fate at the convergence of all these signals or whatever. And I think that's a real, for me, and I think for Arun, that these are the tools that we use still today. I mean, these are the guideposts that allow us to generate hypotheses. And you guys did it back in the day before CRISPR, before Talon even. You were grinding out there, screening probably thousands of clones, a lot of work. So you deserve a ton of credit. And I hope that the younger audience recognizes that as much as all of us old fogies do. Glad that we got to shine a bit of light on that. Before we let you guys go, we have a quick little foray into your kind of science peripheral lives. First, Elizabeth, let's start with you. If you were not a scientist, what do you think you'd be doing with your life?
I would be a flamenco dancer.
Oh my goodness, that is spectacular. spectacular. Put that in the books, or we have not had, I don't know if we'd any kind of dancer, but flamenco. That's real. They got a lot of flamenco in Australia?
Not really. We are definitely a flamenco outpost. However, we do have a Spanish community here, and I grew up learning guitar, and one of the things you obviously learn in guitar is classical Spanish music. So I've always had a love for this, but flamenco is truly unique. It is complicated, mathematical, and very passionate all at the same time. And so
Wow.
I find that being able to take my mind into that world is truly extraordinary. It allows you to think of the world in a different way.
Wow, a lot of parallels to science there. I love it. Finally, either one of you, what's the best piece of advice that you've ever been given, either professional or not?
Well, for me, it was never make important life decisions when you are unhappy.
Well, I know Andrew's advice to the lab probably stands. Everything is important.
Often what I say is a variation of it is everything makes a difference.
Yeah.
Because the question that we get is, does it matter that I use a different base medium? Everything makes a difference.
Matters.
Does it matter that I use I'm not using this molecule, I'm using something different? Yeah, it's easy because the answer is always the same. Because look, a lot of the detail obviously we can't go into, but one of the ridiculous series of experiments that Elizabeth did many years ago was to compare the same growth factors, right, with a different medium base. And what do you find? You find that the differentiations are different. So in other words, when someone says, look, we're using StemPro 34 or something else, does it make a difference? Yep,
yes,
it does. And that's why Elizabeth developed initially the Apple medium, which STEMCELL Technologies made. And
no way, you made that? You must be rich. Yeah. Gosh.
No, but the R Institute might be.
So the harsh reality I have to tell my students, etc., when they sort of talk about this, is I have to say I spend my life doing non-sexy experiments. Everyone wants the big break,
the home run. Yeah, for sure. But at least you got the flamenco in your mind as you're doing all that. You got sexy in your head. But you guys, both of you, this has been such a joy for me, the realization of a conversation I've long wished to have, and I hope we can continue it. We're going to be on your side of the world, close at least, for the ISSCR. If you guys are going to be there, I hope we can throw a couple back and maybe talk about the next thing. There's got to be a fourth thing there on the developmental hemogenesis pathway, which is leaning translational. I'm sure you got your eyes, your targets set on that. But it's really been great talking to you, and thank you guys so much for sharing with us and our listeners today.
Been our pleasure.
It was a pleasure. Thank you very much.
All right, that brings us to the end of this episode, a great one in my opinion. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. You're going to have to do a pretty good job to beat the two we had on today, living legends, two scientists that I've really admired and benefited from in my career. Thank you guys so much for listening to this one, and we'll have another in a couple weeks. Until then, take care.